Hello, dear listeners. Assalamu alaikum to everyone. We are studying hormones, and our today's lecture is all about steroid hormones. What are steroid hormones? They are group of hormones that belongs to a class of a chemical compound which is known as a steroid. These hormones are secreted by three glands. We can call them steroid glands, which include number one, the adrenal cortex, number two, testis, and number three, ovaries. And there is also another organ which is produced especially during pregnancy known as placenta. So, the fourth one. All steroid hormones are derived from cholesterol. They are not packaged but synthesized and immediately released. They are transported through the bloodstream to the cells of various target organs, where they carry out and perform the regulation of a wide range of physiological functions. As these hormones are derived from cholesterol, so they differ only in the ring structure and side chains attached to the cholesterol backbone, which contain four rings. The enzymes which produce steroid hormones from cholesterol are specially located in mitochondria and the smooth endoplasmic reticulum. As all steroid hormones are lipid soluble and water insoluble, so they are permeable to membranes and are not stored in the cells. Steroid hormones are carried in the blood complexed as they are not water soluble, so they must be carried in the blood complex to the specific binding globulins. So these globulins are specific for a specific steroid. For example, corticosteroid binding globulin, which will carry a cortisol, which is a corticosteroid hormone. Similarly, sex steroid binding globulin will carry testosterone and estradiol, while in some cases, a steroid is secreted by one cell and is converted to the active steroid by the target cell. For example, we can say androgen, which is secreted by the gonads and is converted into estrogen in the brain. As far as classification of the steroids is concerned, these hormones are often classified according to the organs they are synthesized in. For example, the adrenal steroids, there are two types of steroids. Adrenal steroids, also known as corticosteroids hormones, and sex hormones or gonads. Adrenal steroids contain two more hormones which are known as glucocorticoids and mineralocorticoids. While sex hormones, there are three sex hormones which are released by ovaries and testes. They are known as androgens, estrogens, and progesterones. This distinction is not exclusive. However, because the adrenal cortex also secretes sex hormones sometimes, for example, LB to a lesser extent than do the gonads. And the ovaries sometimes under abnormal conditions may also produce adrenal steroids. But most of the times we can classify these steroid hormones according to this classification. The adrenal cortex produces the adrenocortical hormones, which consist of the glucocorticoids and mineralocorticoids. So, depending upon their chemistry, there are basically five types of steroid hormones: glucocorticoids, mineralocorticoids, androgens, estrogens, and progesterones. While talking about their details, glucocorticoids such as cortisol, it control or influence many metabolic processes, including the formation of glucose from amino acids and fatty acids, and the deposition of glycogen in the liver, or conversion of glucose towards glycogen, which performs in the liver. Glucocorticoids also help to maintain normal blood pressure and their anti-inflammatory and similarly immunosuppressive actions which have which are useful in treating rheumatoid arthritis and also preventing the rejection of transplanted organs. So we can say that glucocorticoids mainly affect metabolism by decreasing inflammation and by increasing resistance to stress. The important glucocorticoid hormones are cortisol, corticosterone, and 11 dehydrocorticosterone and cortisone. There is very little difference between structures of these four hormones. The next is mineralocorticoids, such as aldosterone, an example. It helps maintain the balance between water and salts in the body. Most importantly, it exerts their effects within the kidney. Then comes sex hormones, gonads, or androgen. First of all, we will talk about androgen. They are male sex hormones. The principal androgen, testosterone, is produced primarily by the testes and in lesser amount by the adrenal cortex. So there are two organs which produce these testosterones, testis and adrenal cortex, and especially by the ovaries sometimes in women. They are produced by the ovaries also. Androgens are primarily responsible for the development and maintenance of reproductive functions and similarly stimulation of the secondary sex characteristics in the male. Androgens also have an anabolic function in stimulating the production of skeletal muscles and bones as well as red blood cells. These synthetic hormones have been used by individuals desiring to increase their muscle mass, such as they are also used by athletes, especially, which seek to gain a competitive advantage of their muscular power. Overdosing of androgens have been linked to serious side effects, which can include infertility and similarly coronary heart disease. Then we will talk about estrogens. They are one of the two types of female sex hormones. They are secreted mainly by the ovaries, and in very small amount they are also secreted by adrenal glands and by the testes also. Estradiol is the most potent of the estrogens. Functioning similarly to androgens, the estrogens also promote the development of the primary and secondary female sex characteristics. So we can say that androgens perform almost similar functions in male while estrogen perform same functions in female. They also stimulate linear growth and skeletal maturation. In other mammals, other than humans, these hormones have been shown heat, to participate the production of heat in the body, which is also known as estrus. The ovarian production is very of estrogen is stopped during menopause. Then comes progesterones, which is also known as progestins. They are most important and are another type, the second type of female sex hormone, and are named because of their role in maintaining pregnancy. So, due to pregnancy term, they are known as progestation or progestins. Estrogens and progestins, they both are secreted cyclically during menstruation. During the menstrual cycle, the ruptured ovarian follicle, which is known as corpus luteum of the ovary, produces progesterone. The placenta becomes the main source of progesterone here, without which pregnancy would terminate. As pregnancy processes, placenta production of progesterone increases, and these high doses suppress ovulation, preventing a second conception. So the contraceptive quality of progesterone led to the development of structurally modified progestins and estrogens. There is another type of hormone known as pheromone. What are pheromones? They are widespread among insects and vertebrates. They are also found in crustaceans but are unknown among birds. It means they are not found in birds. These chemicals may be secreted by special glands present in these animals or incorporated in other substances such as urine. They may be shed freely into the environment, or pheromones may be deposited in carefully chosen locations by these animals which produce them. Pheromones are also used by some fungi, slime molds, and algae as attractants in the process of their reproduction. Organisms of complementary reproductive cell types grow or move towards each other. This slide shows structure of steroid hormones. There are more than 50 corticosteroid hormones which are produced in the adrenal cortex by reactions that remove side chain from the D ring of cholesterol and introduce oxygen with the help of some enzymes known as cytochrome P450 enzymes. We can see here in this slide the structure of cholesterol, which has four rings, the three hexagonal, one pentagonal ring in its backbone, while cholesterol is a 27 carbon containing molecule. While during conversion of cholesterol towards steroid hormones, the break occur at among the carbon number 22 and 23, resulting in the formation of cyclopentanoperhydrophenanthine molecule and which contain total 17 carbon molecule. All steroid hormones are originated from this molecule, which is known as cyclopentanoperhydrophenanthine. The structure of corticosteroids. Corticosteroids are a class of steroid hormones which are released by the adrenal cortex and includes glucocorticoids and mineralocorticoids ensemble. However, the term corticosteroids is generally used to refer to glucocorticoids most of the times, named for their effect in carbohydrate metabolism. Due to carbohydrate metabolism, these steroid hormones are known as glucocorticoids. Glucocorticoids regulate diverse cellular functions including development, homeostasis, metabolism, cognition, and inflammation. When we look upon their structures in detail, it's clear that cortisol has two hydroxy molecules at C11 and C16 along with the carbonyl molecule. While if we look at the structure of corticosterone, we can see that there is only one hydroxy molecule at carbon number 11, while the other hydroxy molecule is absent, is eliminated. While in a cortisone, there is a hydroxy molecule along with the carbonyl carbon, but at carbon number 11, hydroxy group is converted to a ketone group. Whereas in the fourth hormone, both hydroxy groups are removed from carbon number 11 and carbon number 16. Rather, a ketone group is present at carbon number 11 instead of hydroxy group. Here is the structure of aldosterone and testosterone. We can see clearly the backbone of a steroid molecule containing four rings. Aldosterone and testosterone, both of these molecules are basically ketone in their chemical nature. Estradiol is an alcohol containing 18 carbon, while estrone is again a ketone. There are both groups, both functional groups. It contain a ketone and a hydroxy as compared to estradiol. In estradiol, we have two hydroxy group at the either sides of the molecule. Here is the structure of pregnenolone. The This molecule is a vital intermediate molecule for the production of all steroid hormones. And the structure of progesterone, a steroid sex hormone. Now we will talk about synthesis of steroid hormones, how these hormones are synthesized inside the body. Steroid hormones, we have already seen in our last slides that steroid hormones are lipid molecules. They are derived from a common cholesterol precursor. The cholesterol is a 27 carbon containing molecule. There are four major classes of steroid hormones: progestins, androgens, estrogens, and corticoids, which contain progestins contain 21 carbon molecule is a 21 carbon molecule. Similarly, androgen is a 19 carbon containing molecule, while estrogen is an 18 carbon containing molecule, and corticoids they are 21 carbon containing molecule. Steroid hormones are synthesized by dehydrogenases. There are two types of enzymes which are used in the synthesis of steroid hormones. Number one, dehydrogenases, and number two, cytochrome P450 enzymes. They catalyze, both of these enzymes catalyze hydroxylation and dehydroxylation oxidation reaction. Eukaryotic cytochromes P450, they are membrane bound enzymes which are expressed the in either the inner mitochondrial or endoplasmic reticulum membrane of steroid synthesizing tissue. A common and important rate limiting step for the synthesis of all steroid hormone is the conversion of cholesterol towards pregnenolone. Here it is important to know that cholesterol is a 27 carbon containing molecule which is converted to pregnenolone, which is a 21 carbon containing molecule. The common branch point for synthesis of progestins. Uh, Progestins, corticoids, androgens, and all steroid hormones. Expression of the side chain cleavage enzyme, which is known as cytochrome P450, it converts cholesterol to pregnenolone and is one of the unique feature of these steroidal cells that participate in de novo synthesis of steroids. In vertebrates, the synthesis and secretion of gonadal and adrenal steroid hormones are regulated by tropic hormones, which are produced from interior pituitary, such as follicle stimulating hormone, luteinizing hormone, and adrenocorticotropic hormone. Mineralocorticoids are also regulated by iron concentration and circulating levels of angiotensin 2. Now, there is an important question that arises here. How the synthesis of steroids are different from that of synthesis of peptide hormones. While peptide hormones they are encoded by specific genes, steroid hormones are synthesized from the enzymatic modification of cholesterol. The regulation of steroidogenesis involves control of enzymes which modify cholesterol into the steroid hormone of our interest, of the body's interest. Thus, there is no gene which encodes aldosterone. For example, as a result, what happens? There are far fewer different types of steroid than peptide hormones. It means that there are many peptide hormones as compared to steroid. There are only certain steroid hormones which are present in our body. So steroid hormone structures are the same from species to species and they does not differ a lot. So while talking about synthesis of the biosynthesis of the steroid hormone, the first important step is the synthesis of pregnenolone. It is the common precursor for all the steroid hormone. Synthesis of all steroid hormones, four types of steroid hormones starts from this molecule. Adrenocorticotropic hormone stimulates this step. And we already have seen that adrenocorticotropic hormone is released by anterior pituitary gland and is controlled, its release is controlled by hypothalamus. This step is the rate limiting step for synthesis of all steroid hormone. First step is the formation of pregnenolone. Here, cholesterol is first acted upon by an enzyme known as desmolase, and a 6 carbon unit is cleaved off, forming the 21 carbon steroid known as pregnenolone. This 6 carbon unit is cleaved among carbon number 22 and 23. In this slide, we can observe that an enzyme which is named as 2022 desmolase starts this cleavage among carbon number 22 and carbon number 23. Here NADPH is utilized and is converted to NADP, where hydrogens are used to complete the valency of the carbon number 22 and carbon number 23. This is an oxidation step, and ultimately, as a result, we can obtain this molecule which is known as pregnenolone. Pregnenolone is an is a ketone functional group containing molecule. Now, this pregnenolone serves as a principal precursor to all the other steroid hormones which are synthesized by the ovary, testes, or other adrenal cortex. It appears that the rate limiting step for the synthesis of progesterone is side chain cleavage of cholesterol by P450 enzyme. Pregnenolone is here then converted into progesterone by 3 beta hydroxysteroid dehydrogenase. So basically this process is a two-step reaction process. There are two reactions. In first step or in a first reaction, the beta hydroxyl group is converted to a keto group by the help of an enzyme known as 3 beta hydrogenase enzyme. 3 actually its name is 3 beta hydroxysteroid dehydrogenase enzyme. And in the second step, another reaction which is performed by shifting of delta 5 double bond from carbon number 5 towards carbon number 4. So after these two reactions, progesterone is made from pregnenolone. It is also important to know that the deficiency in either of the enzyme, whether P450 or this 3 beta dehydrogenase, it have profound effects. The deficiencies of these enzymes have many important effect on the synthesis of all steroid molecules. Actually, in the ovary, progesterone is produced at all stages of follicular development as an intermediate for androgen and estrogen synthesis, but it becomes a primary secretory product during the uh, little phase of the menstrual cycle. The synthesis of progesterone is under the control of follicle stimulating hormone during the early stages of follicogenesis and luteinizing hormone receptor, which becomes sensitive to luteinizing hormone in the ovarian cycle. So, the synthesis of progesterone by the corpus luteum is stimulated during early pregnancy by increasing levels of gonadotropin. Here is the diagrammatic representation of conversion of pregnenolone towards progesterone. Progesterone is further converted into glucocorticoids. It is the same molecule which is converted into glucocorticoid, which especially for example cortisol in order to make a cortisol, or mineralocorticoid, that is to make aldosterone, and or to make sex steroids, for example testosterone, estrogen, or progesterone. So what are glucocorticoids? The major adrenal glucocorticoids, they are cortisol, cortisone, and corticosterone. These reactions are affected by hydroxylation. These specific hydroxylases are monooxygenases. All these enzymes are NADPH dependent, just as we have seen in the synthesis of pregnenolone. This reaction is also NADPH dependent. This pathway shows the conversion of pregnenolone towards progesterone, and then ultimately progesterone is converted to a cortisol, a glucocorticoid. The predominant human glucocorticoid, that is cortisol, it is synthesized in the zona fasciculata region of the adrenal cortex. It is important to note that the synthesis of cortisol involves hydroxylations of progesterone, as first of all cholesterol is converted to pregnenolone. Pregnenolone, a rate limiting step. At second step, pregnenolone will be converted to progesterone with the help of dehydrogenase enzyme, and then this progesterone will be converted to 17 alpha hydroxyprogesterone. So here we can see that it involves hydroxylations of progesterone at 17 alpha carbon, where a hydroxy molecule is attached, the 21 carbon number 21, and carbon at 11th position, carbon number 11, which is 11 beta carbon positions. The synthesis of cortisol is under the control of an interior pituitary hormone, which is known as ACTH, and a negative feedback mechanism in which elevated cortisol level suppresses the release of ACTH, and ultimately this whole process stops. After three dehydroxylase NADPH dependent steps, that is first hydroxylase step as carbon number 17, another hydroxylase step at carbon number 21, and a third hydroxylase step at 11 beta carbon, we get upon a cortisol molecule. Adrenal mineralocorticoids. The dominant human mineralocorticoid is a aldosterone, but 11-deoxycorticosterone and corticosterone also have significant mineralocorticoid activity. These reactions are affected by hydroxylation. These specific hydroxylases, they are monooxygenases. All these enzymes are also NADPH dependent, and the ACTH stimulates the synthesis of all steroid hormones by activating desmolase, so that the availability of pregnenolone is increased. This biosynthesis is as the aldosterone is produced in the zona glomerulosa of the adrenal cortex. So the synthesis of aldosterone involves the synthesis of corticosterone and subsequent hydroxylation and oxidation at C18 carbon to yield the aldosterone. The synthesis of aldosterone is actually regulated directly by levels of potassium and indirectly by the effects of sodium levels and blood volume on levels of angiotensin 2. In this biosynthesis of aldosterone, we can observe that at first step, an enzyme 21 hydroxylase introduces a hydroxyl group at carbon number 21, while changing a ketone group, a hydroxyl group, towards ketone group at carbon number 3. If we compare deoxycorticosterone towards progesterone, we will find two changes: hydroxyl group at carbon number 21 and acetone group at carbon number 3. In the second step, an enzyme 11 beta hydroxylase introduces a hydroxyl group at carbon number 11 and produce corticosterone. And similarly, at third step, in a third reaction, an enzyme known as 18 hydroxylase introduces a hydroxyl group at carbon number 18 and produce the final molecule known as aldosterone, which has a strong mineralocorticoid activity. Now comes the synthesis of sex hormones, that is testosterone and estrogen. The estrogens and progestins are synthesized and secreted primarily by maturing follicles, corpus luteum of ovaries, and the placenta during pregnancy. The predominant estrogen secreted and the predominant progesterones, which is progesterone, the profile of the synthesis of the estrogen remains changing during the course of follicle formation, during which, under the influence of luteinizing hormone, the cells synthesize and secrete testosterone. In this diagram, we can observe that in the first step, the progesterone is converted to 17 hydroxyprogesterone with the help of an enzyme 17 hydroxylase. This enzyme introduces hydroxylase hydromolecule at the carbon number 17, which ultimately in a second reaction with the help of dismolase enzyme, uh, it removes a total 4 carbon containing unit and convert carbon number 17 towards a ketone. This molecule is known as the resulting molecule is known as androstenedione. This androstenedione, it diffuses across the and along with testosterone, both of these molecules they diffuse across the basement membrane and are subsequently aromatized to estrone by the granulosa cells. This androstenedione molecule, which is a 19 carbon containing molecule, it is converted to testosterone with the help of 17 reductase and NADPH depending enzyme, which introduces a alcohol or a hydroxy molecule at the place of a ketone molecule, and testosterone is then aromatized, which is a NADPH dependent step and is converted to estradiol, a 17 beta carbon containing 17 at the 17 position there is a beta carbon and ultimate and completely it is an 18 carbon containing molecule. Here comes another class of steroid molecules. They are known as urinary steroids. The urinary steroids are referred to as 17 hydroxysteroids, for example, cortisol or cortisone, and also 17 ketosteroids. So they are both hydroxysteroids and ketosteroids. For example, androstenedione, which is an intermediate molecule in the biosynthesis of sex hormones. The steroid hormones these urinary steroid hormones pass through the cell membrane of the target cell, and the steroid hormones binds with a specific receptor present in the cytoplasm. This receptor is present in the cytoplasm of the cell. The receptor which is bound to the steroid hormone, that is a receptor hormone complex, it travels into the nucleus together and binds to another specific receptor which is present in the nucleus on the chromatin. The steroid hormone receptor complex calls the production of a messenger RNA molecule, which then encodes for the production of a specific protein, and these proteins are then transported towards their target cells. So this diagram represents the summary of the whole biosynthesis of the steroid hormones and shows the major pathways for the production of glucocorticoids, mineralocorticoids, And precursors, especially, they are given in the red boxes. Intermediates are given in the gray boxes, and hormones in the blue boxes. Excretory products while in the brown boxes. So starting from cholesterol, in the first step, pregnenolone is produced. This pregnenolone 21 carbon is converted to directly to another molecule known as progesterone. This progesterone is then converted to the remaining steroid hormones. Starting from pregnenolone, which is directly converted to cortisol by some intermediate, and ultimately is converted to 17 hydroxysteroids, which is a urinary steroid. While starting from progesterone, we can see that progesterone is converted to androstenedione molecule, which is a 19 carbon containing molecule with an intermediate by passing through an intermediate of 17 hydroxyprogesterone. This androstenedione is then converted to testosterone and then estradiol, which is then finally converted to another urinary steroid known as a hydroxyurinary steroid known as 17 hydroxysteroid.
Now we will discuss mechanism of action of steroid hormones. It means that how steroid hormones act. Actually, steroid hormones in general have nuclear receptors and they act by regulation of gene expression. As we have already discussed that there are two types of receptors, cell surface receptor and nuclear receptor, and steroid hormones mostly interact through nuclear receptors. Steroid hormones, being lipid soluble, can easily pass through plasma membrane and enter the cytoplasm. Here they combine with specific receptors, and the hormone receptor complex translocates to the nucleus. It transfers to the nucleus. The steroid hormone response elements on DNA and regulate transcription of genes. The proteins produced in response to this hormonal stimulation produce metabolic effect on their target cells, for example enzymes and transporters. So the steroid hormone mechanism of action can be summarized as follows. We can say that number one, first of all, steroid hormones pass through the cell membrane of the target cell. Steroid hormones bind to a protein transporter during the transport in blood of lymph. When they have arrived at the target cell, the protein transporter, which must be fat insoluble, so this transporter is repelled by the cell membrane, while the steroid hormone, which is obviously fat soluble, diffuses into the cytoplasm of the target cell. Here the steroid hormone bind with the specific receptor which is already present in the cytoplasm. This receptor bound steroid hormone, that is a receptor hormone complex, travels into the nucleus, towards the nucleus, and binds to another specific receptor on the chromatin. Steroid hormone also diffuses across the nuclear envelope and enters into the nucleus of target cell. Steroid hormone binds to a specific receptor located on a particular gene of target cell's DNA. This binding alters the genetic information within that particular gene present in the nucleus and result in a new messenger RNA formation which is being produced after transcription. This new messenger RNA still be translated into a new protein, that is converted into a new protein, and this messenger RNA is able to form a new type of protein with the help of an altered gene in the cytoplasm of target cell. The new protein or enzyme which is produced causes a specific effect to occur within the target cell. So in this way steroid hormones act on the target cells. The steroid hormone receptor complex, actually it calls for the production of messenger RNA molecule which code for the production of specific proteins. This is the sequence of steroid hormone action. It's a summary. First of all, endocrine gland secretes steroid hormone. Steroid hormone diffuses through target cell membrane. Hormone combines with the receptor molecule which is already present. The receptor is specific and is already present inside the cytoplasm. Steroid hormone receptor complex enters the nucleus and binds to DNA and promotes synthesis of messenger RNA. Then messenger RNA enters the cytoplasm and direct protein synthesis. Newly synthesized protein produce hormone specific effect, and the new protein causes a specific effect to occur within the target cell. So this is the sequence in which steroid hormone act. Now what is hormone receptor binding and interaction with DNA? The steroid hormone function by nuclear receptor complex. We have already seen in the last slide. This receptor become competent to bind DNA. Actually, first of all, this receptor is present in an inactivated form. As soon as the steroid hormone enters the cell membrane, this receptor turns to activated receptor. Steroid hormones enter the cell by simple diffusion across the plasma membrane. The receptor exists either in the cytoplasm or in the nucleus, which is where they meet the hormone. When hormone bind to receptor, a characteristic series of events occur. Intracellular receptor is located inside target cell in the cytoplasm or nucleus and function as ligand dependent transcription factor. The hormone receptor complex bind to promoter region of responsive gene and stimulate or sometimes inhibits transcription from those genes. The mechanism of action of steroid hormone is to modulate gene expression in target cells. Activated receptor. Okay, the mechanism of action. We have mechanism of action by selectively affecting transcription from a battery of genes, from a collection of genes. The concentration of those respectively proteins are altered. Respective proteins are altered here, which depends upon the selection of proteins is made by the hormone receptor complex, which clearly can change the phenotype of the cell. Receptor activation is the term which is used to describe conformational changes in the receptor induced by hormone binding. So the major consequence of activation is that the receptor become competent to bind the DNA. Now activated receptor bind to hormone response elements, which are short specific sequence of DNA. It's a short specific sequence of DNA that is located in promoter of hormone responsive gene. In most cases, hormone receptor complex bind DNA in pair form. The hormone responsive elements, they are made up of mostly inverted repeats, or might be direct repeats, or sometimes everted repeats, at least 3 or 2 to 8 times respectively. Here transcription from the genes to which the receptor is bound is affected. Most commonly, receptor binding stimulates transcription. It's important to memorize that receptor binding, actually, which it is important, the receptor binding, it stimulates transcription, that is the formation of new proteins which act on their target cell. The hormone receptor complex thus function as a transcription factor. This diagram shows steroid hormones molecular action. If we look upon at this diagram, first of all, we can see that we know that most hydrophobic steroids, they are bound to plasma protein carriers. Only unbound hormones can diffuse into the target cell. So once the steroid hormone receptors enters the cytoplasm, the plasma membrane, they are in the cytoplasm or in the nucleus. There are two sites where a receptor could be found in the cytoplasm or might be in the nucleus. So, after meeting with the receptor, a cytoplasmic hormone receptor complex is created. The receptor then the receptor hormone complex binds to the DNA and activates or represses one or more gene, the relevant genes. These activated genes create new messenger RNA. These activated genes create new messenger RNA that moves back to the cytoplasm and start the process of transcription, that is to make new proteins. Translation. Here translation produces new proteins for cell processes at rough endoplasmic reticulum and Golgi complexes. Some steroid hormones also bind to membrane, the plasma membrane receptors that use second messenger systems to create a rapid cellular response. So this is the whole diagram which represents the hormone receptor mechanism and mechanism of action of steroid hormones. There are some important functions of steroid hormones. Steroid hormones plays a major role in carbohydrate regulation and mineral balance, reproductive functions, inflammatory response, stress response, in bone metabolism, cardiovascular fitness, and activates DNA for protein synthesis. As far as the nature of steroid hormones is concerned, there are some functions with respect to particular steroid hormone, specific steroid hormone. For example, progesterone, which prepares uterus lining for implantation of ovum. Similarly, glucocorticoids, mainly cortisol, which promote gluconeogenesis, that is formation of glucose, either from glucose or from other sources, from fats, etc. It favors breakdown of fat and protein to form glucose and perform fuel mobilization. It's also show an anti-inflammatory action. While if we talk about mineralocorticoids, basically especially aldosterone, they maintain blood volume and blood pressure by increasing sodium reabsorption by kidney. We have already seen that major function of mineralocorticoids is the balance of minerals. While if we talk about sex steroid hormones, androgens, they perform development of male secondary sex characteristics and prevents bone resorption. Actually, same function is performed by estrogen in female, androgen in male and estrogen in female. In estrogen, they form development of female secondary sex characteristics and prevent bone resorption. While there is another very important hormone, vitamin D hormone, which we will discuss in our next lecture. It's not a steroid hormone, but its role is very much similar to the steroid hormones. It performs intestinal calcium absorption, promotes bone formation, and prevents phosphate loss by kidneys. So there are some biological effects of glucocorticoids. The glucocorticoids, as the name suggests, that mainly they affect metabolism of glucose. So the major biological effect of glucocorticoids, they are given in this table. We can see some effects of glucocorticoids on carbohydrates, lipids, proteins and nucleic acids, fluid and electrolytes, their effect of glucocorticoids on bone and calcium reabsorption and secretory action on connective tissues, etc. There are some biological effects of mineralocorticoids. We have already discussed them in last slides. Okay, next comes transport and metabolism of steroid hormones. Actually, secretion of all adrenocortical hormones is under the control of adrenocorticotropic hormone. The diurnal variations of secretion of cortisol, we have already discussed in our last lecture that highest values of cortisol are produced early in the morning and minimum values are produced at night. So parallel, they are these are the actually diurnal variations of secretion of cortisol, and they are parallel to the release of adrenocorticotropic hormone from interior pituitary under the influence of CRF. CRF is a neurotransmitter. Cortisol exerts the negative feedback effect on adrenocorticotropic hormone secretion. Actually, the adrenocorticotropic hormone also increases the secretion of aldosterone. So, we can say that the secretion of aldosterone is indirectly related to cortisol. So, the level of aldosterone is also affected by the position. We can say that highest value is of aldosterone is secreted when a person is in upright posture, and lowest value is secreted when a person is laying down. All steroid hormones act through intracellular messengers and increase the rate of transcription. We have already seen in last slide. Approximately 70% of cortisol in blood is bound to an alpha 1 globulin called cortisol binding globulin CBG, or there is another name known as transcortin, while about only 20% is bound to albumin and the rest is free, which is the biologically active fraction. It's important to note that half-life of cortisol is just about 2 hours. The steroid hormones, they are metabolized and inactivated by the liver. The major processes are reduction and conjugation here. Steroids are 21 carbon containing compound and are reduced to their tetrahydroderivatives, which are also excreted as their gluconoids or sulfates in urine. There are some adrenal hyperfunction which cause Cushing syndrome or ectopic ACTH production, hormone production by other malignant tumor, and there is adrenal hypofunction which cause Addison's disease. The ovarian hormones, estrogens, androgens and progesterones, and testicular hormones. This table shows the summary of steroid hormones. Here hormone is given and the tissue of region from where this hormone is produced, and its specific function is given in the third column. Thank you.